Hello, I'm Ertao Wang from CPAMS, Chinese Academic of Sciences. So today I'm going to talk about the agriculture fused by biological nitrogen fixation. To get it started, I would like to introduce our food. So we know we need carbohydrate, fats, protein, vitamins, and fiber. So every day. So protein tend to be particularly issue in our nutrient since the protein are unlike carbohydrate for most part. So the protein need we get enough protein so for human body for healthy. So we know that we want to protein rich food every day. So something we are going to remember nitrogen. Nitrogen is a critical element. So we need some protein rich food, including some meat, milk. So of course, whether you eat meat or plant, all body, so the protein all body all come from plant. So we need plant every day. So where do this is the important question? Where do the plant nitrogen come from? So the nitrogen from plant nitrogen come from the soil mineral. So however, the fields grew over and over again, and the nitrogen soil will become depleted, which will not support plant growth. Will not support the plant to make protein for us. So how to overcome this? So at agriculture system, the overused field can be recovered by application of nitrogen fertilizer. So usually in form of ammonia. So we know that ammonia can be made from molecular nitrogen, which is the most abundant in nature. So in nature, but then we know that the 3 band of nitrogen is very stable. We need to break down these 3 band. This will cost a lot energy, use a high temperature to convert molecular nitrogen to ammonia. So the ammonia was applicated into a soil, plant can uptake ammonia to make protein. So, but this progress take a lot energy. So this is just to show you 1 or 2% of global energy was used to fix nitrogen to fertilizer plant. In addition, we know that excessive use of nitrogen fertilizer will lead to serious air pollution and water pollution and soil pollution. We know that in China, we cost around 1/3 of the nitrogen fertilizer. This indicating that a low efficiency in fertilizer uptake by our crop. So how to overcome this? How to reduce nitrogen fertilizer usage? So please let me introduce our root nodule symbiosis. Root nodule symbiosis is a mutual beneficial association between plant and rhizobia. In this symbiosis, plant can provide organic carbon from photosynthesis. Meanwhile, the rhizobia can fix nitrogen in nodule and provide ammonia to plant and promote plant growth. I would like to mention that most rhizobia can't fix nitrogen outside nodule. So the rhizobia stay into nodule to fix nitrogen. So all this happened in nodule in room temperature, but no energy cost, only use photosynthesis carbon. So just let me show you how important the legume in our agriculture system. Just recently, we found several fields. We did not apply any fertilizer, such as nitrogen, phosphate, and potassium. When we plant soybean in this field, we found that the soybean yield was not changed at all without nitrogen fertilizer application. That do indicate soybean achieve great yield without any fertilizer application. This field was last for 40 years, no any nitrogen fertilizer application. That do suggest the legume can deal with low nitrogen fixation. So we use modern technology and quantified the microbiome of the soybean root and found at least 2 key factors. One is some beneficial microbe cluster, and another is rhizobia. They function together to sustain soybean yield and low nitrogen fertilizer soil. So you can see the soybean can deal with low fertilizer condition and achieve great yield. So this is really important. So I just mentioned in 2022, around 150 million mu was used to grow soybean. So potentially every year we take 1/2 billion kilogram nitrogen putting in 1/2 million kilogram nitrogen into the soil. So this kind of 1/2 billion kilogram can be totally removed or reduced. So I think the next policy or need to do something to reduce this kind of nitrogen fertilizer usement. So this is show you how powerful, how important the legume rhizobia symbiosis in our agriculture system. Since my lab has been working on microbial symbiosis for more than 10 years, we are interested in how do plants form symbiosis with rhizobia? Where do legume rhizobia symbiosis involved from? So more and more evidence shows that there are some progress, some key gene in AM symbiosis was recruited by legume rhizobia symbiosis lately, used for legume plant established symbiosis with rhizobia. Before this, I would like to introduce a little bit for AM symbiosis. AM symbiosis is really Asian symbiosis. You can see 80% of plants can form this symbiosis with macrolide fungi. So it was first described as by German botanist Frank. So in this symbiosis, the carbon flow to AM fungi, and inorganic nutrient move to plants, thereby providing a critical linkage between the plant and root and the soil. Just lately, the research found that phosphate is a major nutrient transferred from AM fungi to plant. Lately, my group together with other group found that the lipid are a major carbon source transferred from plant to arbustic macro fungi. But we know this by direct nutrient exchange was directly coordinated, regulated by host and environment condition. So normally we see this nutrient exchange was regulated by phosphate condition. And the low phosphate condition, we normally see higher macrolide colonization. So just recently we found that one MIP transcription factor we call PHR can associate with promoter of macrolide-related gene and promotes this gene expression. So we can see the low phosphate in soil can promote arbuscle macro symbiosis. Under high phosphorid condition, SPX can function as a phosphate sensor, directly inhibit PHR-associated promoter of macro-related gene and inhibit this gene expression. So we see under high phosphorid condition, low macro-colonization. So under this, we can update the basic concept of arbuscle macro symbiosis. In this mutual interaction, macro fungi can provide phosphate to plants. Meanwhile, plant can provide lipid as a major carbon source to arbuscle macro fungi. So this whole progress was controlled by SPX PHR module. So this PHR SPX module can make this symbiosis adapt to different environment conditions. So this is the basic concept of macro symbiosis, how plant establish symbiosis with macro fungi. But we know that in one key thing, this progress can be recruited by legume for rhizobia symbiosis. We know in legume rhizobia symbiosis, root nodule is key, since rhizobia need to stay in nodule to fix nitrogen. So next, we want to know how to plant form a nodule. So let me introduce a little bit of background. So in legume, the cortical cell can be dedifferentiated to form a nodule primordia. Laterly form a nodule on legume root. But we know that in non-legume, the cortical cell cannot dedifferentiate to divide. So we think this progress, we think the cortical cell in legume and in non-legume could be very different. So based on this, we hypothesize that a unique property of a legume root cortical cell may be fundamentally enable root nodule development. So based on this, we think some K gene maybe have some extended expression in this progress. Basically, we found one gene in this progress play important role in this progress, which is called SCAR. You can see here. The legume SCAR gene in the middle, you can see the promoter of SCAR drive GFP expressed in endodermis and a cortical cell. So the cortical cell laterally can be developed for a nodule. But for non-legume AT SCAR, which only expressed in endodermis, so the two genes is very different, have different expression pattern in legume and non-legume. So next, we found that the SCAR and SCAR-like double mutant show a defect in root nodule symbiosis. You can see here almost no nodule at a double mutant. So to suggest the SCAR gene is required for root nodule symbiosis, we know that SCAR gene is a K development gene. The mutant show short root, so the reduction of root nodule number could impact by root development. So we use specific cortical cell specific promoter to drive a SCAR dominant negative gene to reduce SCAR gene function in cortical cell specifically. So you can see the transgenic plant should reduce nodule formation. So this data indicate that cortical cell SCAR gene is required for root nodule development. Just to remember Philippe Benfei, who discovered 20 years ago for SCAR and SHR gene. SCAR gene is a grass transcription factor which expressed mainly in endodermis, just as I showed you before. This is in non-legume. Normally, scar gene interact with SHR to regulate root stem cell. You can see SHR gene expressed in stele and move into endodermis and interact with scar gene to regulate root stem cell. Just like in legume, we found that SHR gene mainly expressed in stele, like Arabidopsis, but surprisingly, we found the scar gene present in legume cortex cell. You just see here, this is a gas staining experiment, but Arabidopsis scar, a short root not present in cortex cell. So that indicates a short root gene can be expressed in stele, and the protein can move into cortex cell from the stele. So we also found that one region we call region 2 can determine a short root movement. Since in Arabidopsis 1, if we remove this region, we call the AT scar R2, the protein can move into cortex cell. That suggests that during evolution, legume plants lose region 2. This protein can have a bigger mobility, can move into cortex cell from stele. So of course we use a cortex cell specific promoter, try to knock down function of short root in cortex cell specifically. You can see here the root nodule formation was greatly significantly reduced in this transition-like plant. That suggests the protein scar in cortex cell is really important for root nodule development. So now we know that in legume, the stem cell program, SHR gene are present in cortex cell, which will allow cortex cell with a competence for division. During rhizobia infection, the symbiotic signal can active scar SHR gene and protein in cortex cell, which will lead cell division, finally form a nodule. But we know in non-legume Arabidopsis, we see no scar SHR, so no competence for division, so no nodule formation on a non-legume root. But I have no time to show the data. All data show that ectopic expression scar SHR in non-legume could lead to cortex cell division. So that suggests that scar SHR can be used to engineer cortex cell fate for nitrogen fixation in future. So this is how a nodule involved during rhizobia evolution. But we know that rhizobia symbiosis was formed 100 million years ago, and this progress was selected again and again during natural environment. But we know currently the soybean, the pea just grows in our agriculture system in farmland. So these two conditions is very different. So we just think we can further increase the efficiency of root nodule symbiosis. So the concept is I show here. This is wild type soybean. Normally we see the rhizobia can fix nitrogen and feed plant. Meanwhile, the plant can translocate some carbon to rhizobia to fix nitrogen. But this progress was selected during past 100 million. But in agriculture system, we think we can optimize the nodule formation and then slightly increase nodule number and increase nitrogen fixation ability to promote plant growth. Meanwhile, slightly increase carbon translocation to rhizobia. Our collaborator, Yuefeng Guan, just generate a different kind of mutant. You can see here, this is wild type, HG-6, this is wild type. There's some mutant no nodule formation, some supernodulation mutant NAC1 show 600 nodule per plant. But you can see here, no nodule plant growth not so good, but a high supernodulation plant, the plant grows smaller. But we do find some slightly increased root nodule symbiosis or root nodule number can promote plant growth. So we grow this plant in a field. You can see here, this is wild type. The plant grows yellow. So the transgenic plant, modification plant show dark green, which indicates the plant have more nitrogen. Also we just quantified the yield in 3 years, different place. We found that 10 to 30% of yield, soybean yield, increased in these two genetic modification plant. That suggests in agriculture system we can rebalance nitrogen fixation and carbon exchange between plant and rhizobia. So this is great. We show you how legume important for our agriculture system. Also, I show you how we can further improve legume rhizobia symbiosis to promote legume product. But the problem, I told you, this is only happened in legume. But the biology nitrogen fixation is not is absent in non-legume. So everybody wants to engineer nitrogen fixation in non-legume. So in our field, 3 steps was proposed to engineer nitrogen fixation in non-legume. Step 1, we try to engineer of associative the nitrogen fixation between non-legume plant and the nitrogen fixing bacteria. The step 2 is engineer a key regulator of nodulation symbiosis in non-legume. In this step, we have successfully engineered rhizobia perception in non-legume rice. So you can see this is a calcium spiking trace. Normally the rice can't respond to node factor well, but in transgenic plants, this plant can respond to node factor. So that suggests that we can engineer rhizobia perception in non-legume. As I mentioned before, use scar CHR, we can engineer non-legume corticel to divide. Probably in future we can engineer a nodule at non-legume root to host the rhizobia. But a lot of people, a lot of labs also work on this topic now. So step 3 is engineer nitrogenase in non-legume crop. But we know that easily clear that engineer nitrogen fixation in non-legume, it will take long time. But it's already clear that all agriculture has been fused by nitrogen fixation in non-legume already, as I show you. But in future, I hope not so long, not so long. I hope agriculture can be fused by nitrogen fixation in non-legume. So I would like to stop here. Thanks to my lab memory. Thanks for my collaborators. Also, thanks for your attention.